It's my pleasure to introduce our next speaker and awardee, Dr. Jessica Hua. Jessica got her undergraduate degree, a Bachelor of Science at UC San Diego in psychology, and had Elizabeth Plumley as her advisor. She then worked for a couple of years in Rachel Loewy's lab and got interested in early psychosis. She went on to do a PhD in clinical psychology at the University of Missouri, working in John Kern's lab, again, focused on early psychosis, and came to San Francisco to the VA to do her clinical internship and connected with our group and became a MIREC fellow in schizophrenia and has been working as a MIREC fellow until recently, where she successfully competed for a VA Career Development Award known as an ECAP, or Early Career Award Program through the Vision 21. It's a 2-year award that was just introduced. She's one of the first awardees. And in association with that, she joined our faculty as an assistant adjunct professor. And I'm glad to report that she's also now successfully gotten notice that her CDA2, which is a 5-year award that will follow the ECAP, has already been awarded. She has 27 papers, including 16 first author papers. So she's been very productive and prolific. Dr. Hua's research uses multimodal imaging techniques focused on structural and functional brain abnormalities across the illness course of schizophrenia, from the clinical high-risk syndrome to chronic illness. She also specializes in CBT for psychosis and mood disorders, as well as DBT for personality and mood disorders. So she is also a clinician. And with that introduction, I'm glad to invite Dr. Hua to come forward and give her presentation.
I hope everyone can hear me. My voice is partially gone since I have a persistent cough still, but I have a set of cough drops if needed. Okay. Okay, can you all hear me? Okay, never spoken to a mic before, so we're trying this out. So thank you all for inviting me to present today, and thank you, Dr. Mathlon, for the introduction. So today I'll be presenting on mismatch negativity and theta oscillations evoked by auditory deviance in early illness schizophrenia. Research on EEG-based biomarkers is critical, as they can provide insights into sensory, cognitive, and perceptual processes and their underlying neural activity. In fact, biomarkers can serve many purposes when studying disorders, such as they can be prognostic or predictive and indicate risk of developing a disorder. They can be diagnostic and identify a disorder or a condition. They can be related to staging and help to classify the extent of disease processes. They can be prognostic and report disorder presentation and guide clinical treatment. And they can also be pharmacodynamic and help to evaluate the body's response to treatment and intervention. So many EEG-based schizophrenia biomarkers consist of different ERP components. ERPs are small voltage changes in the scalp-embedded EEG signal, and they're time-locked to the onset of stimuli or responses. And because of this, we are able to capture cognitive processes with very high temporal resolution. This deterministic response to a given stimulus is difficult to detect, though, from the raw EEG because it's buried in background activity and noise. So in order to extract the ERP, EEG is averaged over multiple trials, which results in the EEG noise canceling itself out, and the remaining systematic positive and negative voltage deflections are the ERP components. So to illustrate this, on the right is a 10-second snippet of EEG activity with an auditory tone in green presented every 1 second. It is impossible, as you can see, to detect the ERP signal in this very messy EEG activity. Therefore, it is necessary to cut this 10-second snippet into 1-second epochs, lining up the epochs along the auditory stimulus onset. These trials are then averaged, canceling the noise in the EEG. This resulting average response shows the expected auditory N100 ERP component that occurs around 100 milliseconds following the auditory onset. Of note, as can be seen by the differences in the y-axis of the two panels, the N100 amplitude is very small relative to the range of raw EEG amplitudes, and only becomes apparent because of this signal averaging. So what is auditory mismatch negativity? MMN is a negative voltage deflection elicited when a series of repeated standard sounds is interrupted by a discriminable deviant sound. So to illustrate how MMN is measured, so we have in orange is the ERP elicited by hearing frequent standard sounds. This shows a small N100 response right here. And then in green is the ERP elicited when we hear an infrequent deviant sound, which shows a larger N100 followed by a subsequent negativity lasting until about 200 milliseconds. This subsequent negativity is the MMN, but its amplitude is traditionally measured as the peak negativity in the ERP deviant minus standard difference wave. So the green minus the orange wave. And this is also shown as the blue negativity in this MMN scalp topography map. MMN amplitude is largest at the fronto-central midline scalp electrosites. So critically, MMN is elicited automatically and pre-attentively, while participants ignore presented sounds and perform an unrelated visual task. As such, this task is very important in research examining biomarkers in neuropsychiatric disorders such as schizophrenia, because it eliminates influences of attention, motivation, and cognitive ability that can confound other tasks examining neural activity. And additionally, MMN in human and non-human primates is dependent on glutamate transmission at NMDA receptors, with NMDA receptor antagonists such as ketamine, reducing MMN amplitude. So MMN was originally identified in the early 1980s and has traditionally been studied using time domain ERP techniques. MMN peak amplitude reduction is one of the most widely replicated ERP abnormalities in schizophrenia, and it is widely considered to reflect glutamatergic NMDA receptor hypofunction, a leading candidate pathophysiological mechanism in schizophrenia. So EEG is comprised of a mixture of neural oscillations at different frequency bands, magnitudes, and phases over time. On the right are some of the canonical frequency bands measured with EEG. EEG oscillations are modeled as sine waves and can be characterized by the magnitude, so the height of the wave, as well as the phase, so where you are along the curve of the wave. And increased availability of EEG time frequency analytic tools has led to increased interest in parsing these various neurooscillatory frequency bands and phases summed together in ERP waveforms. So two key time frequency metrics that we assessed in the current study are total power which measures the average change in magnitude of oscillations following stimulus onset, and intertrial coherence, which measures the degree to which the phase of oscillations is synchronous across trials. So to better illustrate this, here is an example of oscillations from multiple trials. So prior to stimulus onset, so to the left of that vertical black bar, the peaks and valleys of the oscillations are offset and average out to 0. However, after stimulus onset, so here, the oscillations are phase synchronized, meaning that the peaks across trials line up and that the valleys across trials line up. Thus, there is high ITC and the waves here. Thus, there's high ITC and the waves survive averaging over trials and appear in the ERP. However, the magnitude of the signal from pre to post stimulus is the same across trials. Thus, there's no change in total power. Okay, and next, here's another example to help illustrate this. So in contrast to the previous example, post-stimulus oscillations are out of phase across trials. There's no phase synchrony, ITC is 0, and very little of the oscillations survive averaging, leading to a very flat and negligible ERP. However, the magnitude of oscillations increases from pre to post-stimulus, resulting in a large total power. So time frequency analyses of EEG collected during MMM paradigms indicate that theta oscillations may underlie MMN. Responses to standards on the left and deviants on the right show a significant increase in theta activity to deviants, but not to standards. Importantly, theta oscillations arise from recurrent inhibitory inputs from somatostatin expressing GABAergic interneurons onto pyramidal excitatory neurons. And translational studies have found that pharmacogenetic silencing of these interneurons and administration of NMDA receptor antagonists such as ketamine and phencyclidine disrupt MMN and its associated theta response. So recent schizophrenia research has begun to examine auditory deviance processing using both these traditional ERP approaches as well as time frequency analysis. In this figure, the authors examined auditory MMN in both controls in blue and patients with chronic schizophrenia in green. They found deficits across all measures, MMN, theta total power, and theta ITC in patients with schizophrenia. As such, what does it mean that there are deficits in MMN as well as theta metrics in chronic schizophrenia? Do theta oscillations provide us with a redundant measure of deviance processing, or do they provide us with independent information regarding the pathophysiology of schizophrenia? Some people have argued that they are reflections of the same phenomenon, whereas others have argued that auditory deviant elicited theta metrics may be a more precise and sensitive measure of pathophysiological processes in schizophrenia than MMN. So thus far, I have been talking about MMN and theta deficits in chronic schizophrenia, and research on auditory deviance processing in early illness schizophrenia is critical in order to minimize the confounding influences of illness chronicity, such as prolonged medication exposure, social occupational dysfunction, and medical comorbidities. Previous research has found MMN amplitude reduction in early schizophrenia, and in contrast to ERP approaches, little is known about whether theta oscillatory dysfunction in MMN is present during these early years. So this leads us to our current study. To address these gaps, we assess theta phase synchrony and total power in response to auditory deviance and further examine their associations with and dissociability from corresponding MMN amplitudes. As such, this leads us to three main study questions. One, do early illness schizophrenia individuals show auditory deviance elicited theta oscillation deficits in addition to MMN deficits relative to healthy controls? Two, to what degree are MMN and theta measures correlated? And three, to what extent are MMN and theta measures elicited by auditory deviance sensitive to distinct pathophysiological processes in schizophrenia. Or in other words, are group differences in MMN fully accounted for by theta measures, and vice versa. So 105 healthy controls and 89 early schizophrenia individuals participated in a prior study in Dr. Mathalon's lab. Healthy control participants were without a current or lifetime Axis 1 mental health diagnosis, whereas the schizophrenia group were diagnosed with schizophrenia or schizoaffective disorder and were within 5 years of illness onset. And to assess MMN, all participants completed 2 auditory oddball paradigms while also completing a picture word matching task. The 2 deviant paradigm consists of 2 different deviant types: duration deviants, which are longer than standard tones, and pitch deviants, which are higher pitch than standard tones. And previous early illness schizophrenia research has suggested that variability in MMN findings may partially reflect heterogeneity of the pathophysiological mechanisms, with some schizophrenia individuals showing greater deficits for duration and others showing greater deficits for pitch deviance. So to maximize sensitivity to schizophrenia despite this heterogeneity, we included double deviants, which were both longer and higher pitched than standards. So going back to our first study question, do early illness schizophrenia show deficits in both MMN and theta measures relative to healthy controls? So here are ERP waveforms for MMN amplitude across the 3 deviant types. As can be seen by the negative inflection in the ERP, both groups showed a mismatch negativity effect, with the healthy control group showing a larger negative amplitude than the schizophrenia group in red across all 3 deviant types. This difference is also represented in the scalp topography plots below, where you can see a much darker blue representative of greater MMN for the healthy controls at the 6 frontocentral electrode sites than the early illness schizophrenia group. And here are plots of theta total power over time relative to stimulus onset. So on the top row, breaking this down, are standards, and on the bottom row are deviants. Healthy controls are again in blue, and schizophrenia patients are in red. And these gray bars represent the time windows from which we extracted our data. And as can be seen in the top row of this figure for standards, early illness schizophrenia relative to controls did not significantly differ on theta power. As you can see, the lines are pretty much overlapping. However, looking at the bottom row for deviants, the early illness schizophrenia group showed significant theta total power reduction across all 3 deviant types. And here we're looking at the ITC plot, so the phase synchrony. Same as the previous slide, on the top row are standards and on the bottom row are deviants. As can be seen in the top row of this figure, the schizophrenia group relative to controls showed theta ITC reduction. ITC reduction for standards, as you can see by the separation between the blue and the red lines. And in the bottom row for deviants, the schizophrenia group relative to controls showed a significant theta ITC reduction, specifically for duration deviants, so deviants that were longer than standards. However, they did not show this for pitch or double deviants. So now we know that both MMN and theta metrics show deficits in early illness schizophrenia. To what degree are these metrics correlated? Examining correlations between corresponding deviant MMN and theta metrics in each group, MMN and theta measures were highly correlated in both groups, with MMN more strongly correlated with theta ITC in green than total power in yellow. This suggests that synchronization of the phase of theta oscillations contributes more to the MMN than the increase in the magnitude of theta oscillations in response to deviance. And as MMN and theta metrics are highly correlated in our data, so to what extent are these measures extracted from the same paradigm sensitive to distinct pathophysiological processes in schizophrenia? In analyses of covariance, I examined whether significant group MMN differences from study question number 1 survived after simultaneously accounting for corresponding effects of theta power and theta ITC. As can be seen in this table, group MMN deficits were not fully accounted for by theta, with pitch and double deviant MMN differences persisting after simultaneously accounting for theta power and ITC. And next, I examined the converse, and I looked at whether early schizophrenia deficits in theta were still significant after accounting for MMN this time. Similarly, theta deficits were not fully accounted for by MMN, with duration deviant ITC differences persisting after accounting for the MMN effect. So thus, these ANCOVA results suggest that although MMN and theta ITC metrics are highly correlated in healthy controls in early illness schizophrenia, they do provide independent information from each other. And additionally, we examine correlations between auditory deviance with symptom severity and antipsychotic dosage in our early illness schizophrenia group. MMN and theta metrics were not significantly associated with symptom severity. However, MMN reduction specifically for double deviance was significantly correlated with increased antipsychotic dosage in the early illness group. However, it is impossible to disentangle if this is a true medication effect or whether individuals who are more symptomatic are prescribed higher antipsychotic dosages. And in summary, our findings replicate previous MMN amplitude findings in early illness schizophrenia and also extend them by showing deviant-related theta ITC and total power reductions. MMN and theta metrics were highly correlated in both groups, with MMN more strongly correlated with theta ITC than total power. And despite these strong correlations, covariance for deficits in one did not fully account for group differences in the other, suggesting their sensitivity to dissociable pathophysiological processes in schizophrenia. Such as it could be that MMN deficits represent NMDA receptor hypofunction, and theta deficits may represent somatostatin expressing interneuron dysfunction. And lastly, only MMN reduction for double deviance was significantly correlated with higher antipsychotic dosages in early illness schizophrenia, possibly reflecting effects of medication or clinical severity. So overall, this research on MMN and theta biomarkers can help to better elucidate pathophysiological processes underlying schizophrenia, with more research needed in areas such as examining how they may predict risk of developing psychosis, maybe indices of remission in schizophrenia, and maybe a marker used to evaluate treatment response. I would like to thank the Brain Imaging and EEG Laboratory, especially Dan Mathalon, Judy Ford, Brian Roach, Susanna Fryer, Holly Hamilton, and Sam Abram. Research was supported by an NIMH grant awarded to Dr. Mathalon and my MyRec Fellowship. I would also like to thank UCSF for inviting me to present today, and to all of you for joining me for my presentation. Thank you so much. Questions while I grab a cough drop?
I think we have some time, or a few minutes for questions.
Thanks for a really nice talk, and congratulations on the award. As I understand it, there's a sizable literature of resting state and event-related EEG differences between healthy controls and people with schizophrenia. So could you couch these findings within that broader literature? That is there something unique about these particular measures? Do they differentiate healthy controls and schizophrenia patients better or differently or uniquely?
Oh, that is a very good question. I think the two of them provide different processes. The resting state helps us look at the activity, the connections between the different regions. And these EEG results help us to look at these very basic early processes that are happening very early in time. But I do think they're complementary approaches. And they can really be used together to tell us information about the different mechanisms in schizophrenia.
Other questions? We have one question from the chat, and this is from Dr. Jackson Trick. Did you characterize your subjects by the presence, quality, as it in like demand versus other, and duration of auditory hallucinations?
Did we characterize the?
And are there any relationships?
Yeah, so I looked at, for the clinical symptom severity, I looked at overall correlations between these MMN and theta metrics with overall positive symptom severity, so hallucinations and delusions. And there was no significant correlation between the two. And I also separately looked at them specifically at hallucinations, and there also was no correlation with hallucinations.
Congratulations on the award. Lovely talk. Just wondering, are you pursuing this line of research further? And if you are, or even if you're not, you've shown that these may provide insight into differentiable biological mechanisms. I'm just wondering where you're thinking about taking this next. I mean, at this stage, you've shown some correlation with with the double dissociation, I guess, or what do you call it, double deviance. But I'm wondering, sort of, you gave us this tantalizing hypothesis that one may be SST, and so where do you go from here?
Yeah, I would like to look at this more in the future, especially this is a big learning area for me, the first time I've done EEG research. So yes, I would love to look at this and more. And currently I'm looking at this right now with Dr. Mathlon, looking at it in a clinical high risk population. And yeah, it'd be interesting to see are we seeing that these deviants are predicting conversion to psychosis? Are they related to functioning, since there's been some research showing that it is related to functioning, and we find the deficits in low functioning schizophrenia, but not individuals who are high functioning compared to controls?
Just if there's no one else with a pressing question, I just wanted to, you went by it pretty quickly, and I don't know this area at all, but you mentioned that there's a mechanistic understanding and that there are pharmacological interventions that sort of back that up. So I presume the mechanistic understanding is coming from mouse or rat models. And I'm wondering, is the pharmacological data, is that human data that when you give people ketamine, you alter this? So are there, I guess I'm wondering are there particular opportunities to kind of link the preclinical and clinical mechanistic work?
Yeah, I think there are opportunities to that. And I think one of the big things like we're seeing like in this data right now is that we're seeing that MMN is more highly correlated with ITC. So we're thinking that maybe it could be related to phase resetting, but we're not really sure yet why we're seeing that relationship, what those mechanisms are underlying that. So it'd be really great to look at research, potentially using animal models to see what those mechanisms are that are helping that phase resetting.
I think we're just about out of time. I don't see any other hands raised, so I think we can bring this to an end. Okay, thank you everyone for attending, and thank you to our awardees for their great presentations.